Well, good afternoon, everybody, and welcome to another one of our Mindset Lecture Series. And today I am actually very excited about our guest speaker, someone who I've had the privilege of getting to know this year, and it's Dr. Deborah Mash. Dr. Deborah Mash is an emeritus professor of neurology and molecular and cellular pharmacology at the University of Miami School of Medicine. From 1986 to 2018, she was a professor at the University of Miami's Miller School of Medicine, where she founded the university's brain bank. Dr. Mash has held uninterrupted funding from the National Institute on Drug Abuse for over 25 years, was one of the world's foremost scientific experts in the anti-addiction drug Ibogaine, which you're going to hear all about today. And she is credited with the discovery of the active metabolite of Ibogaine, which is Ibogaine. Now, in 1994, Dr. Mash received the first approval by the FDA to study Ibogaine and conducted phase 1 and phase 2 trials for cocaine addiction. She then opened up the Healing Visions Ibogaine Clinic, which treated more than 300 patients and presented and generated data from 1996 to 2003. In 2010, she founded DemiRx to advance clinical development of the metabolite Ibogaine and is currently the CEO. So this is an amazing example of an academic research going into industry and really using the knowledge that you worked so hard to obtain in order to really make a drug potentially available to the millions of people who will benefit from it. Today, Dr. Mash will be presenting a scientific introduction to Ibogaine for the treatment of addiction, and I know that you're going to enjoy it. So welcome, Dr. Mash, and thank you so much. The floor is yours.
Thank you. Thank you very much for that very concise introduction. This has been a very long journey for me, and it's indeed an honor to present to you today on work that is many, many decades in its totality. So with that introduction, I want to take you through this. When we think about addiction and we think about plant-based medicines, I want to just emphasize in this slide that Mother Nature gives us many different addicting alkaloids, and three of them are listed here. Heroin from the poppies, erythroxyl and coca for cocaine, and nicotine. So here are three alkaloids from Mother Nature that are very addicting. In 2017, I decided, made a plan to go back into my company, DemoRx, precisely at the height of the opioid epidemic in our country. And as I think all of our listeners know, America has suffered greatly from the influx of heroin from Afghanistan into the US and from Mexico into the US, and then of course, the prescription opioid drug epidemic in our country, and then the fentanyl analogs that have come and flooded our shores and are currently a major problem. And altogether, this has led to a large number of deaths and a lot of wreckage of families and a lot of human suffering, and not least of which are the neonatal withdrawal syndrome, the numbers of infants that are born to opioid dependent mothers. It's a very, very sad and disturbing thing. And so I had a little bit of a crisis of conscience in 2017 where I thought I needed to go back in and refinish what I had started many, many years ago. And when we look at currently what's available, the drugs for opioid use disorder, and we put this into context with the burden of disease, this is summarized here, but I want to point out right now, so looking back on my academic career at the number of new molecules that have entered into the approval process, into regulatory review and approval, there really haven't been a lot. And for opioid dependence, naltrexone is an old molecule, an opioid antagonist, which has been used also for alcohol use disorder, developed primarily out of academic laboratories. Of course, it's also used for that indication. But primarily what we have is methadone, which came into the United States and developed through Rockefeller University, and then National Institute on Drug Abuse, and working within the public and private combinations of sectors, was able to repurpose buprenorphine. When I started Demorex originally to work on noribogaine, and I was the chief scientific officer in 2010, the volume of sales of Suboxone were about $300 million. Today, they are well over 2.7 billion, and it's estimated that by 2026, the Suboxone marketplace will be upward of about 4.5 billion. So this shows you the numbers of individuals that have access to Suboxone in our society, and the number of people that are being transitioned to long-term Suboxone maintenance. The cost to our society is large. The US opioid epidemic since 2001 is estimated to be at $1 trillion, and it's likely to exceed another $500 million over the next 3 years. So developing novel treatments for opioid use and other substance use disorders is clearly an unmet need. The other point I'd like to make here in this slide is that opioid use disorder, the way we think of it clinically, is that, of course, it's an acquired disease. If you never go out and use drugs and you don't get addicted, you're not going to suffer from this brain disorder. But why is it so difficult? Why is it that big pharmaceutical companies have not looked to develop molecules for the treatment of addiction? And part of it is that the disorder, this acquired disease, is one that is complicated by many, many factors. So what are you treating? Are you treating the reward deficiency syndrome? Are you targeting your molecule for withdrawal? Are you looking for long-term abstinence? Are you trying to block relapse to prior patterns of use? So we know that when you abuse drugs and you become tolerant to them, there's a neuroadaptation that occurs in the central nervous system. But also what's important is that many of the people who abuse drugs are self-medicating other disorders like depression and or trauma, to name two. So we have a high comorbidity of drug abuse, and specifically opioid use disorder, with mood and anxiety disorders. And it may be that in order to develop a molecule that's really going to be efficacious for addiction, that you need to modulate more than one target to move a complex CNS disease towards health. So that was our guiding hypothesis and our interest in this molecule, Ibogaine. So Ibogaine is an indole alkaloid that comes from the roots of Tabernanthe iboga. This is actually a photograph that was taken by a BBC reporter for me many, many years ago. It was taken in the early 90s when he went into the region of Gabon and Cameroon, and he learned about Ibogaine firsthand from practitioners of the Bwiti religion. And these are some iboga roots shown here. The ethnopharmacology, ethnobotany of Ibogaine goes back over 100 years. And before the development of pharmaceuticals and the expansion of pharmaceutical synthetic and semi-synthetic molecules, most of our blockbuster drugs, of course, came from Mother Nature. This slide summarizes some of the key facts around the history behind the ethnobotanical and pharmaceutical use of Ibogaine. First of all, Ibogaine comes into Europe from Africa, the French-speaking parts of Africa, into France, where it was actually developed under the trade name Lamborine. And it was marketed in France in these low-dose, this is the powder here, Lamborine powder, but it was actually formulated in 8 milligram tablets, and it was used for 20 years in that context until it was regulated by the DEA. The potential of Ibogaine as an addiction interrupter was first described in the 1970s. And this was at the time, of course, when LSD and the psychedelics were gaining popularity. And Ibogaine had made its way from Africa into the US, and some of the underground therapists were starting to work with it. It was then discovered serendipitously that Ibogaine could have this property of being a profound addiction interrupter. But that really didn't advance until later, and the seminal discovery was made by Howard Lutzoff himself, abusing heroin and at the time, and he then developed a company called NDA International, and he patented Ibogaine for 4 different indications: for opioid, psychostimulants, nicotine, alcohol. And I meet Howard Lutzoff in 1992 because he contacts me, where he wants to use some of the research that's ongoing in my laboratory at the University of Miami to promote his polydrug dependency patent. So that became my introduction to Ibogaine. I actually traveled at that time to Amsterdam to see the Underground Railroad of Addicts Helping Addicts, and that's what they called it, an addict helping addict underground railroad, where people were administering Ibogaine in the Netherlands. When I went to Amsterdam and I saw the effects of the drug, I believed that we needed to study it. So I came back to the University of Miami, got together with my clinical colleagues, and we submitted our first application to the FDA, which Dr. Yehuda mentioned in my introduction. Today, Ibogaine is used commercially in clinics offering medically supported withdrawal management in countries where Ibogaine is unregulated. And these clinics, so-called pop-up clinics, are occurring in Canada, in Europe, in South Africa, in New Zealand, the Bahamas, all over Mexico. There are many clinics in Mexico, Costa Rica. So there has really been, over the last 10 years, a huge expansion of people taking Ibogaine outside the United States. My laboratory was first to describe the active metabolite of Ibogaine, and I'm going to show you some slides on that. And in 2019, our company, Demorex, forged an agreement with Atai Life Sciences, which then led to the funding and our approval to conduct a phase 1/2A trial of Ibogaine for opioid withdrawal management in 2021. And I'm going to show you a few slides towards the end of the talk about the trial design. This slide illustrates what we had approved from the FDA. And really, when you think about this, this was 1993 when I first went up in an open hearing with the FDA in front of the Drug Abuse Advisory Committee. At the time, Curtis Wright and others were in that division, and I worked very closely with NIDA to prepare for this meeting. And it really was a breakthrough, because there were very few groups at the time who had DEA Schedule I license, and there were very few groups that had permission to test so-called psychedelic medicines in humans. One was Rick Strausman, who was at the University of New Mexico, and he had DMT, and the other was Dr. Charles Grobe out at UCLA, and he was beginning his work with MDMA. And then it was Ibogaine in Miami. So that was it. We were the big 3. The FDA was extremely collaborative with us. The medical safety officers and the division was very, very helpful in guiding us. And we actually gained permission to go full speed ahead from a low dose starting range of 2 all the way up to 8 milligrams per kilogram. And today we know 8 milligrams per kilogram would likely be an intent to treat target dose for this indication. So had I been able to raise the funding at the time, imagine where Ibogaine would be today. But unfortunately, I wasn't able to fund it. I wasn't able to fund it, although I submitted many grants to the National Institute on Drug Abuse, and I wasn't able to fund it through the public or private sectors from foundation or other types of support. Some of the key facts to summarize then is that we know from not only work that we've done, but also from many published studies that it's estimated today that about more than 10,000 people have taken Ibogaine in various settings. Unfortunately, however, there are no placebo or otherwise controlled studies. There are many highly consistent reports of efficacy that have appeared in published publications. So these are peer-reviewed publications, and the consistent reports demonstrate in open label studies that it is a very powerful way of managing withdrawal symptoms. It also does seem to diminish cravings, and that these cravings are extended over weeks to months. Reports of abstinence, long-term abstinence, are in the literature and reported. And it also seems to have some benefit as a rapid acting antidepressant. Despite that good information, there's also some bad information. So the dark side of Ibogaine is that people have taken Ibogaine in unsafe settings, and there are currently 33 publicly reported deaths in the literature. And there is also some indication that Ibogaine will cause QT prolongation. And QT prolongation is something that regulatory agencies become very worried about, and it's something that can block a drug from being developed in the clinic. I'm going to share more data about this towards the end of the slides. So our dosing, this slide summarizes our current dosing experience with Ibogaine, and this is work comes from our group. Again, from the University of Miami study, we completed 1 and 2 mg per kg cohort study. And when I went off to St. Kitts, when I couldn't fund this in the United States and we did the trial in St. Kitts, we covered the range of about 6 to 14 milligrams per kilogram. Most of the subjects were in the range of 8 to 12 milligrams. We published on a subset of the cohort that were from patient volunteers. There were opioid dependent, about 102 of those in that publication. The second largest group was cocaine dependent patient volunteers, but we also have an additional 86 patient charts and results from polydrug dependency, alcohol dependence, and a small number of people who suffered from amphetamine dependence. The largest case series is this report in Frontiers in Pharmacology, and this is the report that summarizes the results for opioid and cocaine abusers that I showed in the prior slide. And the data that I'll be showing you in a few slides come from this report. So thinking about withdrawal management, if Ibogaine is an addiction interrupter, and if Ibogaine is effective in blocking the signs and symptoms of opiate withdrawal, well, how does that factor in? Well, we know that for many patients, they can't complete a successful detoxification. And this is somewhat surprising, but they simply cannot either taper down on an opiate to block completely withdrawal. And even if you mitigate the acute symptoms of withdrawal, the problem for patients is the protracted negative affect. And it's this post-withdrawal period that triggers people back into a relapse. So most people can get past the acute withdrawal, although not all. But the real problem here is that once a person is abstinent, then they're affected by this protracted negative affect period. Currently, the opioid withdrawal treatments that are available for use include FDA-approved methadone, which is a full opioid agonist. And in 2018, the FDA also approved lofexidine, which is an alpha-2 adrenergic agonist, much like clonidine, which is used off-label for this indication. Methadone, of course, has a black box warning for addiction, and it is abused and has diversion. It's also is a QT prolonging effect of methadone. Lofexidine similarly has problems with syncope and QT prolongation and bradycardia. So these are used, they're FDA approved, but again, they don't address the post-acute withdrawal period. Today, the current new player is dexmedetomidine, which is a new molecule that's under current development and for this opioid withdrawal indication. But other than that, this is what we have available. So why is it so difficult to complete detoxification? And why is it that people relapse? So this is one of my favorite slides and one of my favorite papers. And people interested in this topic, I encourage you to read this. This is by Christopher Evans and Dr. Cahill, and I believe it's really a seminal paper that explains what's going on here. So when you think about, why do people abuse opioids? They abuse them because they love the euphoria. Not everybody gets a euphoria from opioids. I myself get very sick on opiates, so opiates for me are dysphoric. So I would never abuse them. But other people take the opioid and they just feel normal. They love it. They love the euphoria, and then of course they start to get into this cycle of use, and which leads to not only the intoxication period but also the dependence. Because opioids in general are short acting, you're kindling. You can almost think of this as sort of kindling this reward pathway in the brain. When you go off of opioids, you then have this withdrawal syndrome, which can be short acting over a series of days, but then you have the protracted withdrawal syndrome. And over time, days, weeks to months, as life stress comes back in, or you get in an environment where you remember using drugs, you then trigger the craving, which is induced by the memory of the high and the relief of that anxiety and the idea that I'm going to feel better if I do just one more hit of heroin, if I take another vodka martini, if I smoke another cigarette. So this is this pathway to relapse. So how does the Ibogaine fit in? Assuming that Ibogaine is effective as an addiction interrupter and that it blocks withdrawal, why would this have a protracted effect that exists outside the acute detoxification window? Well, the pharmacokinetics of Ibogaine may help explain this in part. So we were involved, as I mentioned, in the demonstration of the metabolic pathway of Ibogaine. And as you can see in this slide, Ibogaine is converted to noribogaine. It's actually O-demethylated on the 12th position. And this occurs through the activity of liver enzymes, specifically cytochrome P452D6, which is a major pathway for many CNS drugs. So many antidepressants, for example, go through this pathway. And if you look at our little molecule here, this part of the molecule to the left looks a lot like serotonin. So we immediately would imagine that noribogaine might have some serotonergic activity. Ibogaine is cleared from the blood. The half-life of Ibogaine is very short acting. It's only about 5 to 6 hours. So in the process of one day, you've already cleared the Ibogaine. But look at the noribogaine. Noribogaine is a very long half-life, and we know that the half-life of noribogaine is anywhere from 24 to 30 hours on the average from studies that we've done with noribogaine as the sole agent in human patient volunteers. So that has been confirmed. And actually, if you look in urine of people who've been treated with Ibogaine, you can detect noribogaine in the urine out to the longest I've looked is about 3 weeks. So it has a very long duration of action of noribogaine compared to the prodrug parent molecule Ibogaine. So that may help explain some of this. So assuming that as you take a dose of Ibogaine and Ibogaine overlaps with the peak of acute withdrawal symptoms, depending on whether you've been abusing heroin, which is a short-acting opiate, versus methadone, which is longer acting, you'll have peak symptoms anywhere from really about 24 to 36 hours. With fentanyl and buprenorphine, it can be even delayed up as long as 4 hours. So the Ibogaine initiates the process, And then noribogaine is probably coming online and helping mitigate the diminishing symptoms over time. And it's really estimated that the full withdrawal syndrome lasts out to about 1 to 2 weeks. And that's not taking into account the protracted abstinence syndrome that I described in the prior slides. So if you look on the internet and you read, the open label experiment, this vast experiment of Ibogaine in the clinics that I described earlier, demonstrate people endorse the fact that Ibogaine blocks withdrawal symptoms. And it does so very abruptly after administration. Patients immediately report that they don't have any withdrawal symptoms. And we were able to confirm that with physician ratings of withdrawal symptoms after cessation of opioid use. What's also interesting is that we were able to measure changes in depression scores, and patients were able to complete this transition to abstinence. In St. Kitts' study, it was amazing to us because, of course, we had very little information then, and there was nothing in the published literature. But what was fantastic for us to observe, and what our counselors observed, was that during this period, because we had them in residence for about 12 to 14 days, that the patients didn't leave. They didn't split. And if you look at split rates from people who go into treatment who are early in recovery, many of them don't complete their 30 days in treatment. But these patients post-Ibogaine were motivated and willing to complete the inpatient stay. These data show the rapid antidepressant efficacy and changes in Beck's depression scores. And this was sustained even at 1 month follow-up. And we saw similar, albeit the endorsement of depression symptoms and cocaine dependence was somewhat less than we observed with opiates. But again, the same antidepressant efficacy. And this was very marked, especially in our subjects who spoke to us in the days immediately following Ibogaine. So during the Ibogaine period, they were under full medical monitor. But then when they were released back into the treatment environment, they would come and they would tell us, Dr. Mash, what's in me today? I feel amazing. I feel really good. So this was the low dose noribogaine in the blood, we believe. We used the craving measurement that was developed by Johns Hopkins University, which is the heroin craving questionnaire now. And we also use the same assessment for cocaine craving scores. And what I'm showing you here are 4 factors broken out from that scale. One being a negative mood state, desire, intent to use the drug now, lack of confidence in the person's ability to quit, and expecting positive benefits of drug use. Yeah, I really want to go out and get high right now because I know I'm going to feel better. And what we were able to demonstrate across the board was that patients were not craving to go out and use. And of course, they told us this. They said, I don't feel like I need to go out and get high right now. This is the first time. Every other time I've gone into treatment, I've wanted to go back out and get high. I had to fight to stay in treatment. I wanted to leave. I wanted to go back out and get high. And this was again, early after detoxification. So this was the patients giving us the endorsement. So we know that with repeated drug use, opioid dependence develops, and there is a learned association, e.g. a memory of the high, with relief of aversive withdrawal state. And that's what I showed you in that prior slide from Christopher Evans. So these data then gave us, enthusiasm, if you will, that maybe we had a therapeutic method of care that not only was effective in withdrawal management, because there's other drugs that are available for withdrawal management, but it also had these beneficial after effects on mood and craving. What about the Oneric effects of the drug? Well, I'm a neuropharmacologist. So the phenomenology, when we were first working on this, my thought was maybe we want to disentangle the hallucinatory effects of Ibogaine from the beneficial after effects of Ibogaine. And of course, the hypothesis was that we would do this by shifting gears and focusing solely on noribogaine as the lead molecule and not on Ibogaine. Now, I argued that. I said, we don't need the visions. Why do we need this visionary state? And what is the visionary state of Ibogaine? Well, I've never taken Ibogaine, so I don't know. But what the patients tell us is that it's different than psilocybin and LSD. It's different than MDMA. It's not the empathogenic state that's reported with MDMA, and it's not like a DMT experience. So Ibogaine sits in a box by itself. But what the patients reported to us was that it was very much an intense lucid dream. And depending on their metabolic status, whether they were a fast, slow, or intermediate metabolizer of the drug, their visionary experience would last anywhere from 4 to 8 hours. And then when the noribogaine curve reached a C max, in other words, when the maximal concentration of noribogaine was in blood, the visions would abruptly cease. And the patients would go into a deep phase of cognitive introspection. And then by the next morning, by that evening, the next morning, they were completely out of those effects of Ibogaine, and there was no other persisting effects. Well, what did the patients describe? So in order to try to capture these data, we went to Anthropology Techniques, and this was working with a colleague who's named on the 2018 paper. And we used what is called a semi-structured elicitation narrative, and we coded the content. And this just summarizes sort of the top things reported across all narratives. One was that the patients suggested that they felt that Ibogaine was useful for their drug problem. They had been given insight, that they recognized that they needed to become abstinent. There was also some spiritual content that comes up, as with other psychedelic medicines. But the interesting thing for this was they really weren't willing to repeat the Ibogaine experience. So unlike other psychedelic medicines, Ibogaine is not a recreational drug. But is there a benefit to the visions? Well, my therapist said yes. And when I said no, we're going to get rid of the hallucinatory aspect of this treatment, focus on the noribogaine. We know noribogaine doesn't cause hallucinations. Okay. Today, as I sit here, I've changed gears, because we know from the renaissance of psychedelic medicines and the great advances that are ongoing today in many laboratories, including the classic work done by Dr. Yehuda and others with MDMA, is that these drugs are very powerful, and they give us a window on the brain and behavior, and how they're working. I can say for Ibogaine, I don't know. As a scientist sitting here today, I don't know, but obviously Ibogaine must be doing something to turn on metaplasticity in the brain that has a lasting effect. And we know that some of the chemical effects of Ibogaine may be ketamine-like, because there is an action on NMDA receptors. It is an NMDA blocker like ketamine. But also the way the patients describe the experience, the Oneric effects, or these waking lucid dream-like effects, seem to bring about a very focused engagement of the patient with the content of the Ibogaine experience, and that this may be in some way engaging the frontal lobe. But there's much more work to be done, and this is purely speculative. So what would I say as a pharmacologist? Well, I would say as a pharmacologist that the polypharmacy actions of Ibogaine and noribogaine are probably explaining the efficacy, the open label efficacy that we and others have been able to demonstrate, and that the self-tapering effect of the metabolite explains the easy transition to abstinence that patients report, and that we've been able to measure in open label clinical setting. A little bit about noribogaine. So I mentioned to you about noribogaine having a long half-life, and what I'm showing you here are data from Demorex, where we conducted a study with Ibogaine as the sole agent. And you can see, if you look at the elimination half-life, here I'm showing you a dose, increasing dose from 60 to 120 to 180 milligrams of Ibogaine, that the elimination half-life is anywhere between 30 to, in some cases, upward. You see the range is shown here of 48 hours. So very long elimination half-life. And there are proof of concept studies, non-clinical studies in animals that demonstrate that Ibogaine is contributing to the efficacy of Ibogaine in human subjects, including published work from my laboratory on demonstrating that mice treated with Ibogaine have diminished withdrawal symptoms and signs in a naloxone precipitated withdrawal model. Ibogaine has no rewarding effects. Again, low abuse liability here in conditioned place preference study in rodent. It's not self-administered. This is an unpublished study conducted for Demorex by Susan Schenk. It has no kappa dysphoria, no NMDA discriminative stimulus, different from Ibogaine, as I mentioned about ketamine. It will decrease cocaine and morphine self-administration in rodent models, work done by Stan Glick. We demonstrated in Demorex that it blocks nicotine self-administration in rats, and elegant work from Dorrit Ron and her collaborators at the Gallo demonstrating that it decreases alcohol consumption in rats as well. And these are work from Amir Rizvani this is one of my favorite slides, actually. Amir Rizvani did this work in alcohol-preferring rats, and he demonstrates that Ibogaine dose-dependently decreases alcohol intake in rodent models. Ibogaine's binding sites, what targets are we hitting? Well, it's an interesting mix, and these are Demorex and other published data from the literature, but I just highlight the blue here. Ibogaine is a potent serotonin reuptake inhibitor, anywhere from about right around 50 nanomolar, so this would be similar to Paxil. It's also a biased kappa agonist, which is it has an interesting atypical opioid pharmacology. So it's acting on the kappa, so it's a kappa and a serotonergic. And then also Stan Glick has, pioneered this idea. He was the one who made the first observation about Ibogaine acting on the alpha-3 beta-4, and these are the neuronal nicotinic receptors in the habenula that put the brakes on dopamine-mediated reinforcement. And he and others in our own lab, we have demonstrated that, and of course, 18-MC, 18-methoxycoronardine, the Ibogaine analog that is currently being advanced by MindMed, acts on this same target. And Ibogaine actually has a higher affinity than 18-MC at this target. And it, again, as I mentioned, Ibogaine has no dwell time in the MK801 channel, so no action on glutamate. So interesting pharmacology with 3 very, very relevant targets. So the polypharmacy mechanism of action of Ibogaine is also augmented by the long-acting metabolite Ibogaine. And we are currently very excited about this, and it also has led us to consider pursuing Ibogaine not only as an adjunctive therapy after Ibogaine detoxification, but also potentially for use in recently abstinent alcoholics. I want to briefly, in closing, make a few more points for discussion today. Those of us that are working with psychedelic medicines and are interested in these medicines because of their psychoplastogen-like effects in the brain, because this engagement of the frontal lobes, or thinking about, again, what I had in the prior slide, the change on not only on the neuronal synapses, but also on consciousness and conscious thought, can help engage in sort of the rewiring of the brain and help people to unlearn maladaptive behaviors. But there is a whole nother line of thought now where groups are looking to develop novel platforms of compounds that are not hallucinogens. And going back in time down memory lane with Dr. Mash, well, we did that too. And so back in the late 90s, together with a very talented medicinal chemist, Simon Mbuanagala Afange, who worked with me for many years, we developed what were called azapinoindols, and that's the molecule shown here on the left. We actually had 2 full composition of matter patents. I'm not going to talk about the other one, but I want to introduce the seminal work coming from David Olson and his group. This was a paper published, a non-hallucinogenic psychedelic and analog with therapeutic potential of Ibogaine called tabernanthalog, and the molecular structure is shown here on the left. Here's our compound on top of it. Here's his compound on the left. And David is the co-founder of a company called Delex, which is a platform company that is working on a pipeline of non-hallucinogenic compounds. They have really should look at this. If you're interested in this topic, follow Delex. They just did a Series A $70 million round. They've assembled an incredible group of scientific and clinical collaborators. Kurt Rasmussen left the National Institute on Drug Abuse, the Medication Development Division, to join Delex. And there's a lot of activity and a lot of enthusiasm here. And David is a very talented young investigator. What I'd like to point out here is when I looked at this molecule myself, and I kind of stared at it for a while, and those chemists who are on the call can look at this with me. If you notice, this is the nitrogen, the 5-ring nitrogen of Ibogaine. Mostly when you do Ibogaine, this would be the serotonin part of Ibogaine here to the left. And this was that tropane part that I showed you before. Well, they flipped the molecule around. So this part here would be over here, you see, and this is their nitrogen here. So left, right, and top and down. So if you just take the molecules, flip it left and right, and point the nitrogen down, you will see a very similar relationship of tabernanthalog with our compound series, which was published a long time ago. So there's this is something to watch. There's going to be a real advancement along these non-psychedelic pipelines. The dark side, I'm going to close on this and lead to the trial, and then we can open it for discussion. But the dark side of Ibogaine is that I mentioned all these underground open label use of Ibogaine and people taking desperate people who are desperate for some kind of treatment to break their intractable cycle of abuse are going into unsafe settings. And this, of course, has led to Ibogaine-related deaths. And this is a wonderful, a really well-crafted review article by Professor Corkery on this topic. And he summarizes what was the current state of the literature up until a couple of years ago. I myself published a paper with Matthias Luz, where we were invited to give an expert opinion editorial to this journal, and this article is now online as of June 2021 of this year, where we try to explain and evaluate the toxicity of Ibogaine with these case reports that are in the literature. As I mentioned, adverse events in the cohort study that we conducted at the University of Miami in St. Kitts were mild. So we did not have any Ibogaine reported deaths. We did not have torsade de pointe. We did not have any ventricular ectopy in our study. But other people have. And so there are 11 published articles reporting a total of 12 patients that had torsade de pointes, which is a life-threatening arrhythmia and a very serious thing that we cannot see. So the point that we make in our review is that the findings of torsade are usually, typically, almost always associated with higher, very high doses of the drug, taken from unknown or questionable sources. So in other words, we don't know the purity of the compound. And the adverse events are associated with multi-drug situations, so people that may have other drugs in their blood as well as other concomitant meds. A little bit about our clinical trial. So during the COVID, we were a little bit slowed back, but we are now fully into our study in the UK. So Demorex was very fortunate in working with MHRA, which is the regulatory equivalent of the FDA in the United Kingdom. And they basically allowed us to go forward to bring Ibogaine back into GCP, regulatory review. And this slide illustrates where we are. Basically what they allowed us to do was to do an adaptive design where we're doing a dose escalation arm in healthy volunteers and recreational opioid users. And then once we collect the safety data and of course the cardiovascular risk and the pharmacokinetics are what the regulatory agency wants to see because of the adverse events that I showed you in the prior slide. So in order for us to get regulatory permission to go forward, we need to be able to demonstrate that this drug can be given safe. And basically with all of the data that we showed to the regulators, they said pretty much go back into the clinic, demonstrate what you've already shown us, and we'll allow you to go into phase 2 efficacy arm of the study. And the efficacy arm of the study will allow us to look at the effects of single dose oral Ibogaine at the maximal tolerated dose or the intent to treat dose level on various efficacy endpoints and safety endpoints while patients are under full medical monitor, but then also following out long-term. And working together with the Thai Life Sciences, we plan to develop a digital therapeutic to track this long-lasting efficacy of Ibogaine in this cohort study. Just to follow up, and this is my company hat now, our company is positioning 2 products for US development. One is Ibogaine, as I've explained in the prior slide, specifically for opioid withdrawal symptom management in a fully medically monitored environment, and to promote abstinence, transition to abstinence, and to promote harm reduction and to improve healthcare outcomes. We know that the QTC is our main risk profile, and that's what we're working to mitigate. And we currently have, as I mentioned, MHRA phase 1/2A ongoing approval. Ibogaine, we're moving towards developing it in collaboration with our US colleagues for alcohol use disorder. And I'm really excited about this and hoping to be in 2022 in front of the FDA with our GCP, our clinical plans. And in this case, we would be using low-dose Ibogaine in oral doses, and we will be developing it towards repeated oral dose for this indication. Again, there is a QT prolongation risk, but we know the safety margins are going to be favorable here. The difference with Ibogaine and Ibogaine is that Ibogaine is not metabolized, it's glucuronidated and cleared. So in this cohort use, the indication for use, this will have some benefits. And we plan, as I say, to go into an FDA meeting very shortly. So Demorex is advancing 2 non-addicting molecules for the treatment of addiction, and I hope to be in the clinic not only in the UK but in the United States for alcohol use disorder very soon. And I want to thank all of my colleagues and collaborators. There have been so many people that have worked on this for so many years, and I'm extremely grateful. Thank you.
Well, that was really amazing. Thank you so much for that. I'm going to turn this over now to Lauren. We have some unbelievably great questions from listeners, and I think we're going to have a lively discussion. Lauren?
Yes, thank you so much. Yeah, I'll just get started because we have a lot of questions coming in. Our first one is from Dr. Salsitz, who is an addiction medicine provider and educator. So I think he's going to come up and ask. Oh, here he is. Yes. Dr. Salsitz, are you able to unmute yourself?
Yes, can you hear me?
Yes, thank you.
Yeah. Hi, Dr. Mash. Thank you for that very comprehensive talk. I've been an addiction medicine specialist for a number of years and worked extensively with methadone and now with buprenorphine. And, I think the general consensus is that the goal of using buprenorphine or methadone should not be for withdrawal management, but should be for maintenance. And
Yes.
the literature is very, very clear that 90+% of people will relapse over time.
Mhm.
So when you compared the different medications, and on the last slide, non-addictive medications, I would say that somebody on methadone maintenance or buprenorphine maintenance doing well, functioning well, is not addicted to those medications, is physically dependent but not addicted, and should stay on them indefinitely. And the big mistake people make is trying to get off early and then relapsing. So whether Ibogaine will provide the type of long-term indefinite protection against relapse and craving, I guess is something that we'll have to see as time goes by.
And what Dr. Salsitz is mentioning is very much the consensus of our key opinion leaders in the United States and Canada today, that everyone should be placed, or the majority of patients, let's say, should be placed on medication-assisted therapies like methadone or buprenorphine. So the question is, stay on opioid for a long term. And I think for some patients, indeed, that will probably be the clinical judgment and the best indication for that person. But for other patients who don't, and families who don't want to continue on opioids, who want to try to make a transition off of opioids, can we have an alternative? And some patients, Ed, and I think you would agree with me, would, benefited from naltrexone, but transitioning them off of opioid to naltrexone was not easy for all of them, right? So they weren't able to make that transition. We know that we can transition people with Ibogaine to naltrexone, and many did very well. And indeed, for some of our intractable opioid users, we said, you can't just fly without a safety net, you need to go on naltrexone. I mean, we told people to do this. Go back to your doctor, go back to your addiction psychiatrist and discuss this. But many people, Ed, want to come off, and people have, spoken to me over and over again and said, Dr. Mash, I want to come off of bup, and I can't. I taper down off of buprenorphine and I get down to 2 mgs, and the dysphoria, I'm climbing out of my skin. I can't taper off. The withdrawal dysphoria following a full kappa antagonist is kind of brutal for some of the people. So, whether you stay on, but if you want to have a transition to abstinence, if you want to try to come off, pain patients too, who may have been transitioned to bup, who may want to come off and have a higher, functionality, may be an alternative for addictionologists such as yourself to use, especially if we have noribogaine as an adjunctive.
I agree. The more alternatives we have, the better. The only problem, Dr. Mash, is that many people want to get off, quote unquote, methadone, buprenorphine, and naltrexone because there are a lot of myths and misconceptions. So as long as they have the full correct information, that's okay. But many people are getting off for reasons that make no sense and then relapse and unfortunately have poor outcomes. But thank you very much.
I know there's some discussion today also about using buprenorphine for other drug substance use disorders, including cocaine and alcohol use disorder. I must tell you, I don't see it, but that's me.
Thank you for a wonderful discussion. I'm sure there'll be more. It's very challenging. I guess we'll jump into another question for now. We have Dr. Resnick up.
Hi, thanks for allowing me to participate so I can ask this question directly. So I'm just kind of reintroducing myself into the psychedelic world, I guess. I have a pain and palliative care background as a psychiatrist, and then an addiction background. But really was introduced to the psychedelic world in residency at NYU, where they were studying psilocybin for early on for really more for cancer patients, and now I'm learning more about psychedelics in this burgeoning field, and want to kind of just understand basic terminology. I think I'm getting more familiar with it. So wanted to know what was meant in that last novel drug by the term non-hallucinogenic psychedelic. Is that indeed, in other words, something with sort of the psychedelic aspects that are not hallucinogenic, just as it says? I mean, can you specify?
Yeah, he coined, he's a clever guy, Dave Olson, and I like his work very much personally, but the psychoplastogen idea, right?
Mm-hmm.
So can you count? So assuming that, we, as a pharmacologist, you have a target. You remember cloned, you weren't born then, but when I was young, we were just cloning. I mean, I was mapping receptors when they weren't cloned. That's how old I am, okay? I'll give you that. So, one target drug, right? We wanted one target drug. Now we know the brain is complicated. There's more neurons and neural connections in our brain than there are stars in the Milky Way galaxy. So if you're going to treat a complex CNS disorder, you're modulating a circuit, the connectome, right?
Mm-hmm.
Okay. So neuroimaging has given us that window on the brain. So these molecules, maybe what they're doing is they're acting on growth factors. And one for addiction would be, glial derived growth factor, right?
Mm-hmm.
And, just like NGF, using, the serotonergic growth factors versus the dopamine growth factors. Well, you can't just regulate those in the brain. If these do that, I mean, this is where microdosing comes in too, right? Mm-hmm. So then you can start to tweak those circuits and normalize tone, neurotransmitter tone.
Right.
And then maybe start to get some healing, and then, do neurons that fire together, wire together? Do you see an augmentation of behavioral interventions? How about that?
Interesting. So that's kind of on a biochemical neurotransmitter, neurocircuitry level, but just sort of from a nosology, phenomenology level, the idea is that hallucinogenics maybe are a negative or unnecessary, to the extent that the psychedelic experience is therapeutic, that the hallucinatory part of it maybe is something to separate out as not being as therapeutic. Is that the idea?
Yes. Got it. Yeah. Mm-hmm. And I mean that I was one of those people. Mm-hmm. I was that person. I said, when we were in St. Kitts and doing the studies, and I was away from the psychiatry group, we had psychiatrists there too, and visiting psychiatrists and therapists and everybody with the people. You took an intractable user, somebody who had been in rehab, what Ed said, everybody's in rehab 6, 7, 8 times, and they fail and fail and fail and fail. And now here they're down with Ibogaine, and they would come in and they'd be in the group, and we would put the papers on the wall where they could write down their aftercare plan, and they were white, because they were, they were stabilized, but they were still neurotoxic. So nothing going on here. Mm-hmm. You give them the Ibogaine, they come out of the Ibogaine, 2 days later, the pages fill up. So here was this, I can't make change, now I can make change. So, I mean, that's not me. I'm a receptorologist. So mm-hmm. I'm not good at this, but I think the tools for you, my people, my therapists, the psychiatrists, and they were skeptics, said, this is doing something fundamental, transformative to behavior.
Right.
Do we know what that is? Do we know? I mean, that's going to be your generation to know.
Interesting.
Go get it.
Thank you.
What a way to end. Unfortunately, our webinar ends right at 1, so we're about to disconnect. But thank you so much for coming.
Sorry,
I think, no, there's a ton of questions, and I wish we had another hour.
Oh my God, I talked too long. I'm really, thank you for all these wonderful questions. And you may anybody who wants to talk to me, please email me. I will answer your question by email. So you may give my email out to everybody, and Dr. Yehuda, and to all of you who helped me today, and Ed for that good key opinion leader comment, which is very important and an important question. And Seth, I wish you all the success.
Thank you.
Thank you, Rachel.
Dr. Mash, you know, there's a discussion about this lecture that's going to take place at 5 p.m. Lauren has the link if you don't have it and want to be a part of it. We do that after all of our lectures. And I think you will capture these questions for discussion because they really are excellent. This was so stimulating, so interesting, and so challenging because, you know, you're disrupting the way that people are treating a very important problem. And I can't wait to keep track of this work and see where it goes.
All right, Lauren, invite
Thank you
me.
again, and thanks to
I'll show up at 5 o'clock.